There we go. Awesome. So I'm the first of three Eriks you're going to be hearing from today. And what I'm here to talk to you guys about is an integrated software environment for DNA and RNA nanotechnology design. So you kind of saw a similar slide of this in Andrew's talk just now, where we kind of have this vision for nanostructure design, where you as the designer come up with a design, you build it in some sort of design tool, whether that is Sean Douglas's famous CAD Nano tool, or whether that's one of the newer kind of second generation tools that we've come up with recently for building DNA nanostructures. Let's see. Yep. Okay. So the idea is that once you have this design,
just shorted?
yeah, it's all in there.
We try unplugging and plugging back in. And where's this one headed to? It's loose. Maybe. Maybe it was loose.
Okay. Are we good? Are we going to be stable this time? All right. So, as I was saying, once you get to your simulations, you can start to identify defects. You can start to identify structural properties. You can start to identify modifications you'd like to make to your design. And the idea then is you can take this back to the design phase, redo your design, begin the simulation again, and then finally you take that to the experiments and hopefully it works. And perhaps then you share it with the rest of the community who will use it for their own applications, and you can use it for the applications that you envision your structure for. And so one question that I often get from people who are like hardcore experimentalists is why should we bother using these computational models in order to optimize our DNA structures? And I'd argue there's kind of three main points for the reasons you should be using computation models. Number one is that they give you this mechanistic understanding of molecular interactions that you really can't get from nanoscale experiments. And this is just because of our resolution for identifying what's happening at the nanoscale is really limited. You can't just look at it with a microscope. It's too small. And so in doing this, you can not only understand the interactions, but you can also perform experiments in the simulations that you otherwise would be impossible, either through applying forces to your structures or through perhaps turning on and off interactions that will let you understand the ways that different intermolecular interactions give rise to the final properties of your structure. And so our grand vision for why we want computational models is to not only identify the defects at the molecular scale, we'd also ideally also be able to predict the yield and to characterize the properties of the final structures that you're going to be working with. I don't know why that came in third, but the answer is that they're also higher resolution. So what the Schultz group works with, which I'm a part of, is we work with a coarse grain model of DNA and RNA called OxDNA. And what this is it's a one bead per nucleotide coarse grain model. It was empirically parameterized from biophysical properties and structural characterization of DNA strands. And it's been used kind of in everything from basic biophysics of how DNAs apply under pressure, all the way up to the simulations that Michael Matthews here in the audience has done, where you're looking over 1 million nucleotides in a single scene. So this is why doing these coarse grain models is really important, is because if you want to look at structures at the DNA origami or multi-origami level, this becomes quickly computationally intractable if you want to do a fully atomistic model. So this is a really cool model, but the problem is that when I started my PhD, there was not good tools to work with the model. And so that's kind of what I've spent the last 4 years doing, is basically taking this super cool model of DNA dynamics and then building out things around it. So the first thing, I see we have lost all the GIFs. This could be a problem. So that's supposed to be a GIF showing our molecular visualizer and editor OxView. Basically, this is a super easy to use web application, which is just drag, drop, intuitive editing interface, so you can build structures as well as visualize simulation results without actually having to go through a very laborious process of creating visualizations. And not only is it great for assembling components that came from maybe different design files, but it's also great for doing algorithmic assembly, which in those nanocrystals with millions of nucleotides I showed in the previous structure Misha's been over here writing scripts that let you copy and paste the individual monomers over and over again to build up these supramolecular lattices and then start the simulations in a very time-efficient way. We also wrote a Python library for analyzing the OxDNA trajectories, and since we control both the visualizer and the analysis tools, it was really easy to couple these together and take output files from the Python library, drop them into OxView, and get things like per nucleotide features, such as the RMSF of each particle, or order parameters that vary over the course of a trajectory. In this case, looking at a molecular engine that opens and closes under chemical influence. And in this case, we can look at, if we apply forces, how the structure closes with time. And in each case, we're just building this up with a library of Python functions that made it very easy to build and simulate designs. Another thing that I really wanted to emphasize is that in the DNA origami community, a lot of us really work on the proof of concept side of things. We basically say, we can build a structure with the following properties. But what a lot of people would like to see is more collaborations and interactions with outside the field with people who may not know how to design a DNA structure, but would really like to use the properties of nanoscale positioning to ask maybe some biological or technological questions with them. And so we introduced a website called nanobase.org where designers are able to upload and share their design files to make this more accessible to not only the DNA community to build larger, more complex designs based off of the intuitions of previous researchers, but also to allow the rest of the community to access designs and then use them for their own application purposes. So for all the DNA nanotechnologists in the audience, if you have designs you'd like to upload, it's a super easy form to fill out, but if you have any questions about preparing things for simulation, feel free to reach out to me and I'm happy to walk you through or help with file format conversions to get it all ready to go. So the last thing I want to show as far as what we've done is what we came up with just in the last week. And so this is basically taking it all together. There's a new version of OxDNA which has these Python bindings which lets you start and analyze simulations basically on the fly from a Python notebook. So you can start a simulation, you can analyze the simulation using the Python library that we wrote, and then Misha did some JavaScript wizardry, and you can even just pull up your OxView visualizations inside the notebook as one integrated simulation environment, which is just going to, at least on the perspective of structural DNA characterization, is going to be awesome as far as iterating through designs. So what do we learn from doing this? Number one is that in building off of the OxDNA model, the first thing is that web browsers are awesome. They give you this very great containerized environment that everybody has on their computer. And so if you can make an app that runs in a web browser, basically everybody can use it and you don't have to worry about installation problems. The second thing is to really build on the open source community. So not only make your apps open source so others can build off of you, but look at what's available. We've had some really great successes by building off of the power of NumPy on the Python side and off of Three.js, which is a JavaScript 3D visualization library on the JavaScript side, which really made these apps extremely performant, and we're able to load those million nucleotide structures and still have them, render nicely inside a web browser. Next thing is to work closely with your experimental colleagues. They have questions that they want to ask with the simulation, so listen to them and also use them as your beta testers. They don't know your code, so if they sit down and click on something, watch what they click on and make sure that something intuitive happens that first time they click on something so they are really in tune with how to use your apps. Third thing, and this is when you're preparing for simulations, one of the steps that can be the most difficult is relaxing structures. And so OxView has this nice built-in relaxation algorithm that really helps prepare things for simulation, and it's also really great from the design perspective because it lets you kind of zoom in on an individual unit you'd like to optimize, and then zoom out from the nucleotide to the helix to the bundles of helices levels, and then look at things both from the 2D and then into the 3D orientation. And finally, is to work at the resolution that works for your application. I mentioned that OxDNA is a nucleotide level model. And the important thing here is that nucleotide level is good enough for doing kind of these structural DNA nanotech things. If you need to go higher resolution because you're really interested in individual atom resolution, by all means do that. But don't overcomplicate your systems in such a way that you can't, that it makes it too much information for designers to actually work with it. And so finally, I'd like to share like a couple of visions for what the Schultz lab is going to be working on going into the future, and kind of where we see the community going on the like design, simulate, experiment interface. First thing is we'd like to see more algorithmic design for structures to be able to make things that are beyond kind of the human abilities to design things. So we've had great success building origamis. You've seen many presentations of the last day where people designed an origami, assembled it, maybe they assembled multi-origami things. A recent project we've been working on is something called SAT assembly, which is where you encode problems into Boolean satisfiability problems and use that to predict to generate reusable components to build very large macro structures. Second thing is other materials. We have a very coarse protein model built in OxDNA now, but we'd really like to see support for things like magnesium ions, for lipid membranes, and for small molecules that we don't currently have at the nucleotide resolution. And finally, people have talked a lot about yield, and one of the problems with like pretty much any model is that diffusion limited processes are really hard to model. And so what we'd like to see are both coarser and more multi-scale models for the DNA community to be able to actually model the assembly and take well-characterized blocks and model them at a coarser scale than at the interesting interactions between your building blocks that are where the actual where actual problems might come up in your system. So that's the end of mine. I just got to thank everybody who made this possible, and for all the lab members for being great beta testers. Thank you. If you do have any questions, feel free to reach out to me, and I'll take questions right now. Ben.
So I think this is a question for you and for people more generally, and I think you touched on it very briefly, but to sort of go back to the drum that I keep beating is like, what I really want to see is like, there are so many different ways of simulating so many different systems, and everybody sort of has their own thing. And so like, just to take like, I don't know, Rosetta, for example, like how would you like smush like OxDNA with Rosetta, right? Like the work that like people have done so much work creating protein design tools. Like it'd be, I think, kind of silly for you to reinvent that. So like,
Yeah, exactly.
what does that world look like?
So honestly, it ends up being a very boring kind of computer architecture question because you need to have files and systems that will talk to each other, right? So Rosetta is really amazing at fragment assembling both proteins, and I think they do RNA now too, where you can build up these like kind of detailed models, but then how do you take that and then design maybe a DNA scaffold around it? And so right now we're kind of stuck with the PDB as our format for like transferring data between things. And it works pretty well, but I would like to see more work on how do we encode structure. And especially one thing that DNA is really missing are the DNA design tools are really missing are like modifications, proteins, small molecules that are included in the design, but they're not in the files themselves. So it's kind of a reproducibility and a sharing problem as well of how do we get everybody to talk to each other?
I think it's just important to note that there is such an environment like that, and it's been around for a little while. It was developed by Doi in Japan. He got funding from the Japanese government. Problem is, I can't remember if it's called sushi or martini, but and it goes from atomistic to macroscopic, and it's free. You just have to send him an email, and he'll send you the thing back. So if you look up Doi, then and computer simulation, then.
Yeah, we'll have to look into that.
Is there one more question?
Thank you. That's really exciting to see that you have nanobase.org sharing all these different DNA origami architectures. Is there a platform for people to be able to comment with questions so you could make it like a little discussion board for each?
Oh, that would be a very good, like, next time I have an undergrad who needs a summer project, give me a comment for him.
Okay, that would be awesome.
Great. Thank you, guys.